Welcome to the Sink Your Teeth into Sync for Genes webinar. My name is Tom Deneen with Kaufman and Associates, and will be assisting with the logistical support for the Zoom session. We ask that you select the speaker view option located at the top right side of your Zoom interface. This will allow you to see the speaker as they present information. If you need technical assistance during the session, click on the chat bubble icon at the bottom of your screen, and this will pull up the chat box to the right side of your Zoom interface. Please type the issue into the chat box and one of our techs will respond to you. Finally, please be aware that today's session is being recorded. Closed captioning is available by clicking the CC icon at the bottom of your screen. I will now turn it over to Papia Paul.
Thank you. Hi, everyone, and welcome. We appreciate you taking the time to join us for today's ONC Tech Forum session to learn more about the Sync for Genes project. This project so far has been aimed at helping to advance the sharing of genomic data, an area that plays a critical role in helping to advance personalized medicine, improving patient outcomes, and accelerating medical research. So we're excited to share more about the work to date and the progress that we've made in this space. As mentioned, I'm Papia Paul. I'm a public health analyst here with the Office of Technology at ONC. I'm also joined today by Dr. Bob Primus from the Mayo Clinic, whose expertise and involvement has really been instrumental to many of the Sync for Genes activities. And later on, you'll also get to hear from some of our partners at Children's Hospital Los Angeles, as well as Element Informatics, who participated in the latest phase of our project. So if you have any questions today during the webinar about Sync for Genes, we do encourage you to submit them through the Q&A feature in Zoom, and we will do our best to address them to help make the session as informative as possible. Next slide, please. Okay, so getting into it, here's a quick look at what we'll be covering today. We'll be exploring the Sync for Genes project as a whole, getting a sense of its history, objectives, and key accomplishments. We'll then transition into a focused look at phase 5 of the project, which was the most recently completed phase, examining its goals, activities, and results. So we'll also then take a deeper dive into a real-world application with our most recent demonstration site that we hope you all find interesting. So following that, we'll discuss the outcomes of this specific phase, as well as the opportunities and potential challenges that we've identified over the course of the project. And then where stakeholders are really essential to help continuing to drive progress in this area. Lastly, we'll also point you towards some useful resources that we've generated throughout this project. There's been a wealth of knowledge and information that has come through over the years to help get you started. So let's go ahead and get into it. Next slide, please. Right, so as mentioned, we're going to start with an overview of the Sync for Genes project, which has been underway, as I mentioned, for a few years now. It was launched back in 2017 by ONC and initially in partnership with the National Institutes of Health, or NIH, and their health data gathering effort known as the All of Us Research Program. And since then, we've completed multiple phases, each with distinct objectives and accomplishments to enable the consistent sharing and use of genomic data in research and clinical care. Next slide, please. So with input from the genomics community and industry, the Sync for Genes project identified and prioritized a list of challenges impeding the widespread adoption, implementation, integration, and use of genomic data in the clinical and research domains, which includes a number of key stakeholders, some of which you see outlined here, such as providers, patients, laboratories, and researchers, and also aligns with ONC's aims to improve health and care through the access, exchange, and use of data. Much of the early work that's taken place in this project really highlighted the need for robust industry-supported data exchange standards, such as the Health Level 7 or HL7 Fast Healthcare Interoperability Resources, otherwise known as FHIR, and that standard to support the effective exchange and use of genomic data. It also became apparent that existing standards required greater specificity and granularity. So these identified challenges were addressed as project phases. So it involved stakeholder discussions and technical demonstrations that a number of participating organizations throughout the duration of the project. Most importantly, it was recognized that these challenges were interconnected. So each phase tried to tackle a specific challenge that provided a solution for subsequent phases to build on. So the overarching design of Sync for Genes then set out to demonstrate incremental progress across a series of phases, again, with each phase expanding on the accomplishments of the previous ones. We'll take a closer look at each individual phase, but just kind of going through these quickly at a high level. Phase 1 of the project began with a focus on standardizing genomic data. Phase 2 demonstrated the connectivity and exchange of data, integrating genomic data into the healthcare workflow, Then phase 3 further extended these efforts, focusing on the interoperability of genomic data from laboratories. Phase 4 looked at sharing genomic data for patient care, where the focus was on interoperability of genomic data between organizations, and that included patients or caregivers where appropriate. And then finally, that landed us at phase 5, which we'll dive into more deeply later, both the kind of activities, the findings, as well as the outcomes from that particular phase. And it was focused on standardizing genomic variant sharing and interpretation for clinical knowledge at the point of care. Next slide, please. At a base level, to realize the full potential of genomic data, establishing an understanding and agreement on the data to be shared is necessary to standardize it to help ensure consistency and usability. So phase 1 focused on standardizing genomic data involving organizations from various sectors. They pilot tested use cases from the HL7 clinical genomics workgroup domain analysis model and provided a reference for implementers using FHIR for genomics. So in turn, this phase contributed to the successful publication of the Genomics Reporting Implementation Guide as part of the HL7 FHIR Release 3.0, where it's important to note, since then, significant progress has been made since the initial development. Next slide, please. Next, standardized collection and exchange of genomic data are needed, ensuring its usability for researchers, care teams, and patients. So after reaching consensus on the necessary data, phase 2 expanded on phase 1 by demonstrating the exchange and integration of genomic test results at the point of care using that Genomics Reporting Implementation Guide. So 4 organizations, as you can see here, participated during this phase, each with different focus areas, and the goal was to test FHIR resources against various use cases. Ultimately, the feedback that was received contributed to refining the FHIR specification within the HL7 clinical genomics work group. So this phase was able to successfully demonstrate exchange of genomic diagnostic reports using FHIR, and also importantly identified challenges to larger scale integration of genomic data into health IT. Next slide, please. So as the primary producers of genomic test data, laboratories play a crucial role in exchanging clinical genomic results. So next, we need to ensure standardized genomic lab test results data, allowing for its integration and use. So phase 3 demonstrated the ability to share standardized genomic data generated by laboratories in the healthcare setting while expanding the adoption of the FHIR clinical genomics specification. So this phase presented a really valuable opportunity to test the specification in laboratory settings. The outcome was the sharing of clinical genetic reports that could be integrated and consumed into EHRs, and a new specialized human leukocyte antigen reporting implementation guide using FHIR shorthand. So this phase also helped identify gaps and generate insights for HL7 to further refine the FHIR standard. Next slide, please. So next, phase 4 operationalized the findings from the first 3 phases to develop application programming interfaces, or APIs, focused on sharing genomic data for patient care. So the use of APIs can enhance patient care by facilitating the exchange of genomic information and knowledge among individuals in clinical and research settings. So this phase aim to develop and test the health IT infrastructure for interoperable sharing of genomic data with individuals, in addition to knowledge support using the standardized FHIR APIs. Demonstrations that were carried out during this phase helped to showcase electronic genomic data exchange between organizations and data receivers, such as patients or caregivers. This resulted in the development of a proof of concept API to deliver genomic variant results to healthcare providers, and also created a prototype app which provided a user base to the FHIR API. Next slide, please. Okay. So finally, we arrive at phase 5, which is the culmination of our work so far. We focused on basic variant interpretation as a test case for standardizing, integrating, sharing, and disseminating information, providing that real-world feedback necessary to enhance the use of genomic data. So we aim to improve access to variant annotation data to assist in the clinical interpretation of genomic variants for more informed decision-making. And as I shared earlier, we did recently complete phase 5, and we'll be taking a closer look here in a bit to the phase 5 activities, findings, and outcomes. But I did want to take a moment just to quickly highlight the phase 5 final report, which was just published this month on ONC Tech Forum's webpage and can be accessed at the URL listed here. Next slide, please. Okay, so as discussed so far, so each Sync4Gene space has contributed significantly to our understanding and ability to utilize genomic data in healthcare, from the initial standardization of genomic data in phase 1 to the integration of this data in healthcare workflows in phase 2, the exploration of laboratory generated genomic data in phase 3, and the development of APIs for sharing this data in phase 4. Now I'll hand it over to Bob, who will delve deeper into our phase 5 work.
Thanks very much, Papia. My name is Bob Freemuth. I'm from Mayo Clinic, and I have had the privilege of being part of the Sync4Gene's program for several phases now as a technical director and a subject matter expert. Could I have the next slide, please? Thank you. So as you just heard from Papia, each of the phases of Sync4Gene's focused on a different aspect of genomic data sharing. Phase 5 built on the work of phases 1 through 4, and it explored the capabilities and challenges related to exchanging not only the genomic results themselves, but the annotations that are used to interpret those data. The goals of this phase, of phase 5, were to test the existing technology and infrastructure that supports genomic data and annotations. This includes the use of public genomic knowledge bases and to gather input from domain experts regarding the practical exchange and use of those data. Phase 5 aimed to explore each of these aspects in the context of use cases that included providers and testing labs, which were the focuses of phases 3 and 4. And it did so by defining a use case that included the use of genomic data at the point of care. So as in previous phases of Sync4Gene's, 2 parallel activities were undertaken. A demonstration project that tested the technical infrastructure and standards, and the engagement of a panel of experts that discussed at greater depth key topics that were identified by a similar panel in phase 4. Next slide, please. The expert panel that was convened for phase 5 included the study team and 8 members that provided a wide variety of domain expertise, which included testing labs, physicians, and researchers that focused on both inherited disease and cancer genomics. It also included informaticists that develop standards and implement them as part of the infrastructure used in clinical environments. The panel from phase 4, the previous phase of Sync4Gene's, identified many challenges related to genomic data sharing, which were grouped into 6 broad areas shown here. The phase 5 panel dug into each one of those areas to identify key gaps and opportunities. Next slide, please. The 6 topics that were identified by the panel are listed in the left column on this slide, with summary findings shown on the right. There's more data available online in the Sync4Gene's phase 5 final report if you would like to read more about each of these. The panel recognized gaps in standards and technologies that are currently used to exchange genomic data and knowledge. To avoid proliferation of standards, the panel recommended focusing effort on the continued development and extension of existing standards rather than the development of new standards to fill those gaps. In particular, the panel members identified a need for improved harmonization between the specifications that were produced by HL7, which operates in the clinical EHR space, and those that are in development by the Global Alliance for Genomics and Health, or GA4GH. This type of work would require high levels of coordination among standard development organizations, and that would certainly improve interoperability and enable more seamless data exchange, but that level of engagement across SDOs is higher than what currently exists. The interoperable standard specifications are also necessary to enable lower barriers to adoption and support broader integration within implementations. The panel also discussed the need for the development of more standards-based content, including the description of genomic variations and the annotation data that describes knowledge about the impact of those variations. Many existing knowledge bases capture genomic variations in discrete or semi-discrete forms, and simple variations are often more able to be represented discretely than complex structural variations. And as a result, there are some gaps in our ability to unambiguously represent those types of variants in computable forms. Secondly, the annotations that summarize knowledge about the impact of variations, which are used by labs to interpret a patient's genetic test results, are frequently recorded in unstructured or semi-structured text. Capturing more content as discrete data would improve our ability to access and use those data computably, but that requires both robust standards, as mentioned in the first point here, as well as knowledge-based curation activities. The panel also discussed the pace at which genomic standards are being implemented and adopted in practice. The panel recognized the barrier to adopt standards can be very high, due both to the resources and the expertise that are required. To help lower those barriers, the panel recommended the development of an environment that potential adopters could use to learn about and evaluate genomic standards. Currently, no such environment exists, and it can be a significant effort to create one locally. The development of a sandbox environment for genomic standards would provide a place for those adopters to explore example data and code and to share lessons learned from their implementations. The panel identified challenges related to the infrastructure required to support clinical genomic data at scale. Most of those challenges related to the hardware needed to handle the volume and complexity of genomic data, and the systems required to deliver up-to-date interpretations of genomic data on demand. The panel concluded that while these challenges can be significant, they are dependent on local environments and are best addressed by local decision-making and development that tailors local solutions to needs. The panel also included several clinical providers who recognized that the use of genomic data within clinical practice can be a challenge for patients and providers that have not been trained in genetics. Clinical decision support, or CDS, can help address this challenge by providing guidance and information to help with decision-making. Although the panel did acknowledge that implementing genomic CDS can be a barrier in and of itself. Nonetheless, education is key, and the availability of genomic knowledge in a more computable form, such as mentioned in point 2 above, would improve the ability to find and deliver the right information at the right time. Finally, the last point on this slide, the panel discussed the need for robust training, education, and support, which is required to enable the standardized representation, exchange, and use of genomic data and knowledge. This education spans the technical teams that must implement genomic standards, the providers that create the content and use the data, and the patients that receive the results of their testing and invariably look up information to help them understand how their genes might impact their health. Next slide, please. In parallel with the panel discussion, Sync4Gene's Phase 5 included a demonstration project that explored the current and emerging technologies that support the use of genomic data and knowledge dynamically at the point of care. In this project, CHLA and LMU Informatics developed a proof of concept application for genomic CDS. The app retrieved a patient's genomic test results using the GA4GH variation representation specification and annotations for those results using the GA4GH variation annotation specification. It then delivered those data to a provider through an HL7 FHIR interface. Now, at a high level, this demonstration project evaluated three key aspects: the ability of GA4GH specs to represent genomic data and knowledge, the degree of harmonization between GA4GH specs and HL7 FHIR, and finally, the emerging technology called Operations that makes the HL7 FHIR APIs more convenient to use for a given use case. I'm going to stop here because you'll get to see more about this in just a few minutes when we play a video that shows you the results of the demonstration project. Next slide, please. Before diving into that project, though, I wanted to summarize just a few of the main recommendations from Sync4Gene's Phase 5. The first recommendation is to focus on the development, enhancement, and harmonization of genomic data standards. While it is sometimes easier and faster to develop something new, interoperability will be improved if fragmentation is reduced and effort is dedicated to making existing standards more robust and aligning the specifications so data can be transformed from one to another more easily. This last point, harmonization, is outside the typical scope of most standards development organization activities. And so it is an effort that is usually overlooked and frankly is often skipped altogether. Secondly, we recommended the development of a sandbox environment that would help implementers learn about standard specifications and work through challenges associated with mapping data and testing code. This environment would help provide a place for the community to discuss, to test, and evaluate proposed solutions before they decide to implement and adopt those solutions in production. Thirdly, was to recommend the support for the implementation and adoption of genomic data standards, which would lead to increased use of genomic data to improve health and for research. A variety of activities could fall under this umbrella, including technical connectathons, educational workshops, and other types of training and system demonstrations. Lastly, and in part as a natural outcome of accomplishing the 3 points above, improvements in our ability to represent and exchange complex genomic data and knowledge will enable the use of those data as part of CDS. The combination of more robust data standards, more structured and standardized genomic data, and lessons learned from institutions that already have mature genomic CDS implementations could help lower the barriers for new adopters. Next slide, please. And with that, what we would like to do now is transition to a pre-recorded video that was prepared by our demonstration site. You'll see Shrikar Chamala and Bob Dolan from CHLA and LMU Informatics, respectively, walk through the use cases and the application that was developed as part of this phase of the program. So with that, if we could show the video, please.
Greetings to all. We warmly welcome you to the presentation of Phase 5 for Sync4Genes, where we will delve into a standard-driven methodology for sharing dynamically annotated genomic variants. This presentation will be delivered by myself, Shrikar Chamala, and Dr. Dolan, who had the pleasure of jointly leading this exciting effort. I will be presenting project overview, and Dr. Dolan will be doing the rest of the talk. Overarching goal of ONC Sync4Genes is to standardize the sharing of genomic information, particularly using FHIR. In this project, we have effectively intersected patient-specific genetic variations with knowledge encoded via GF4GH variant annotation method, drawing from databases such as PharmKB, ClinVar, and Civic. The dynamically annotated variants identified through this process are then returned utilizing FHIR Genomic Operations. The HL7 FHIR Genomics Reporting Implementation Guide, or FHIR Genomics, provides FHIR representations for an array of genomic data structures such as variants, haplotypes, and variant annotations. This allows for a standards-driven communication of both simple and structural variants, germline and somatic variants, pharmacogenomic star alleles, HLA typing, and other findings originating from sequencing, chip technology, and cytogenetic analysis. The type of annotations supported by this standard include diagnostic implications, such as a specific variant being associated with a particular disease, therapeutic implications, which might involve the presence of a variant predicting a treatment response to a certain drug, or presence of a haplotype suggesting altered pharmacokinetics of a particular drug. Molecular consequences, for instance, the presence of variant indicating potential loss of function of the gene in it resides. Through these, FHIR Genomics enables comprehensive and precise genomic data communication. Prior to this current work, there has been efforts in the past in harmonizing GF4GH and FHIR Genomics. A portion of the April 2022 GF4GH variant annotation model is shown in this upper left, whereas portions of FHIR Genomics are shown in the lower right. For example, the GF4GH therapeutic efficacy statement profile has been harmonized with the FHIR Genomics therapeutic implication profile and GF4GH pathogenetic statement. In the interest of time, I will not be spending a lot of time on this slide. There is a publication that we recently came out, which can provide a detailed information about FHIR Genomic Operations. Overall, FHIR Genomic Operation extends the FHIR Genomic standards and basic FHIR search capabilities in order to simplify developer access to potentially complex and voluminous data structures. Now I'm gonna pass on to Bob Dolan, who's gonna do the rest of the presentation. Thank you very much.
Thank you, Srikar, for that background information. I want to go in now to a little bit more detail about some of the activities, findings, and outcomes achieved under Sync4Genes Phase 5. Before I do, I want to just revisit this kind of high-level 50,000-foot diagram of the main intent of the project, where we sought to take patient variants and dynamically intersect those variants with knowledge, particularly knowledge encoded using the GA4GH variant annotation specification, and then return those results to clinical applications using FHIR, but more specifically FHIR Genomics Operations. We did this by enhancing the reference implementation, and we delivered the dynamically annotated patient variants to 2 apps, we'll take a look at those apps. But before we look at the apps, these are the main use cases that were driving the work. Number one, clinicians want to view up-to-date genetic implications, including germline cancer screening and germline pharmacogenomic screening. We had another group of clinicians that wanted to filter and prioritize potentially causative variants as part of a rare disease diagnostic workup. So we used the same pipeline, but we wound up creating 2 different apps to show how the dynamic annotation of variants could be used to serve both of these purposes. The way that we approached the work was to enhance the reference implementation. We previously had ClinVar and Civic and PharmGKB knowledge encoded in here in a locally defined schema, but we replaced that with ClinVar, Civic, and PharmGKB knowledge that was encoded per the GA4GH variant annotation specification. We also added a couple of additional annotation pipelines. One, we added a molecular consequence annotation pipeline, and another one was the population allele frequency annotation pipeline. We then extended the previous mapping that we had done a year or so ago, where we had mapped various aspects of GA4GH annotation model into FHIR. And here we also mapped the GA4GH molecular consequence profile into FHIR. We then had to make some revisions to the operations because we made some modifications to the internal data structures. And we then surfaced up the dynamically annotated variants in these two different applications. Number one is the face sheet application that shows the ACMG screening and the PharmGKB screening. And the other application is a application where we can show molecular consequences and disease pathogenicity to support more of the rare disease variant discovery pipeline. And I actually want to show you the face sheet app in a little bit more detail. So let's take a look at the face sheet app in action. So let's select the provider. Let's go ahead and select the patient. And what we'll see here is a typical face sheet summarizing the patient's problems and medications and allergies, to which we've added this widget in the bottom left, which summarizes the results of genomic screening. And in this application, we're doing ACMG screening shown here, where we have associated this patient's variant with cardiomyopathy because we found an entry for this variant in ClinVar. And we provide some additional information in this box here that you can click and further explore the variant. And likewise, we do pharmacogenomic screening for all the CPIC level A drug gene interactions. And then what we do is we take those interactions and we apply them to the problem list, the medication list, and the allergy list. And the way that we apply them to the problem list is we have a little knowledge base behind the scenes that says, look, if the patient has one of these diagnoses, go ahead and just surface one of these other screening findings if it's possible that that genomic finding could be an etiology for the condition on the problem list. So here we have a patient with dilated cardiomyopathy thought due to viral myocarditis, and yet we discover that the patient has a variant associated with a hereditary form of cardiomyopathy. And as my old program director used to say during residency, many of these patients that actually have thalassemia and present with a microcytic anemia are misdiagnosed as having iron deficiency anemia and inappropriately treated with iron. And so here we're basically trying to address that problem by surfacing up alternative diagnoses where we find genetic indicators that might be contributing or might be causative for something on the problem list. Likewise, we go ahead and we look for identified pharmacogenomic interactions. And if we see that there's one of those medications on the medication list whose metabolism might be affected, we flag that. And clopidogrel is particularly interesting in that there's no great biomarker that tells you whether or not the clopidogrel is working appropriately. So even in these patients that have been on clopidogrel for some period of time, the identification of a patient being an intermediate or poor metabolizer of clopidogrel could be clinically relevant. And so we flag those. And likewise, in addition to flagging medications, if the patient has a substance on their allergy list where there's an ingredient that overlaps one of our drug gene interactions, we go ahead and flag that as well. So that's the face sheet app that demonstrates how we deploy dynamically annotated annotations to clinicians to meet the needs of clinicians just wanting to know the latest, greatest information, screening information for their patients. In summary, what we were able to do under this project was to successfully demonstrate an open source pipeline for the sharing of dynamically annotated genomic information where the knowledge was encoded using a GA4GH standard, but the annotated variants were then delivered using FHIR Genomics Operations. We found that by decoupling the update of knowledge from the reporting of the sequencing results, our pipeline enables the most current annotations to be presented to clinicians. Out of all of the various lessons learned, the one that I thought would be most interesting to share with you is that I think most people would intuitively understand that different types of variants might be easier or harder to automatically associate with knowledge in different knowledge bases. And of course, we found the same thing. But interestingly, we also came up with a number of bioinformatics strategies that can greatly improve automated annotation fidelity. And that'll be the subject of a future report. So anyhow, I want to thank you for your time. I want to thank you, Srikar, for the great collaboration in this project, and ONC for sponsoring the work. Thank you very much.
Thanks very much to Srikar and Bob for the demo that you just viewed there. We hope that by taking a look at that recording, you now have a better feel for what was done as part of the Sync4Genes Phase 5 demonstration project, and in particular, why it's important to have data in structured and discrete forms. This demonstration also served to show how intentional harmonization and alignment across standard specifications can improve data flow, as you saw it flow out, in this case, from a couple of public knowledge bases through their little bit of an analytic engine there and clinical decision support, and into a clinical-based use case through a FHIR interface. So we'll now conclude this presentation with a few final comments that will summarize the outcomes from Phase 5 and then generalize to some broader themes. So if I could have the next slide, please. Next slide, please. Thank you. Sync4Genes Phase 5 built on the work that was done in prior phases of the program. It focused specifically on the ability to retrieve and use genomic data and knowledge from public databases in the context of a clinical use case. Standards from GA4GH and HL7 were used to represent the structured and semi-structured data from those knowledge bases, and the ability to capture data and transform it between those standards was evaluated. A panel of experts was convened to form and extend the work that was done in earlier phases, and through those discussions, many gaps and opportunities were identified. And then finally, and I'm just going through this quickly because much of this is review based on what we've already discussed here this morning. Finally, the outcomes of Phase 5 were synthesized with the results of Phases 1 through 4, and some strategic recommendations were developed to inform potential future work in this space. One of the primary messages throughout Sync4Genes has been a need for improved interoperability that is achieved through the alignment of standards and supporting of broad adoption. Next slide, please. Throughout all phases of the Sync4Genes program, a number of challenges and areas of opportunity have been identified. And while it's easy to get lost in those details, a number of recurring and foundational themes have arisen, and where concerted efforts are needed to drive improvements in genomic data sharing. Those themes are listed on this slide. We'll run through them briefly, but they'll sound pretty familiar to you by now based on what you've already heard. The first is related to the strategic development and adoption of genomic standards. We've specifically in Phase 5 looked at standard specifications that have arisen from the FHIR and GA4GH organizations, the former being the language of EHR systems, and the latter having roots more in the research space. Each of those development organizations, standards development organizations, are working to develop standards, and there is both opportunity and risk related to complementarity and overlap. So the alignment between those is really key. That really gets to the second theme that has arisen, which has to do with the support and coordination for implementing and integrating those standards. In this case, within this demonstration that you just saw, we saw data flowing from knowledge bases, which was structured and represented in GA4GH formats, and ultimately delivered through FHIR interfaces. And one of the slides that went by a little bit quickly towards the beginning of that demo was one that showed arrows that connected these two things. And it's the dirty work that occurs within those arrows that can be very challenging and that can require a lot of really careful coordination and negotiation between the owners of those respective standards. The third point here is related to education and training. And as has been mentioned previously within this presentation here today, this is a recurring theme that has come out of Sync4Genes, and that is the need to really support the education of both the technical staff that are tasked with developing and implementing these standards, as well as the consumers of genomic data so that they understand how to use it appropriately. Of course, across a use case like the one that you just saw, if the technical staff and the generators and users of the data are not all on the same page, things can happen which are not intentional. And we can end up munging data in ways that is not appropriate. And so this type of education and training and the development of these standards really hinges on multidisciplinary teams that are talking actively to each other throughout the whole process. And then finally, the last point on this slide relates to challenges that impact the adoption, implementation, integration, and use, not necessarily at the local level, but across industry. A lot of these standards are incorporated into vended software. A lot of them are developed upon locally, and everything needs to talk together effectively in order to achieve interoperability. Now, each of the areas that are called out here on this slide can be addressed by a combination of local effort and collaborative or consortia-based work, such as that which occurs within standard development organizations. Through the work that was done by the Sync4Genes program, we've been able to identify opportunities where organizations like ONC really could have a positive impact across these spaces. Now, by focusing on targeted issues within these key areas, Sync4Genes has been able to pave the way for more efficient and effective genomic data sharing, ultimately improving patient care and accelerating medical research. But of course, much work remains to be done. Next slide, please. And with that, I will turn it back to Papia.
Great. Thank you, Bob, for that overview of the progress that's been achieved in phase 5, along with the Sync4Genes project as a whole. And just to echo some of the comments made, there's been substantial progress made in advancing genomic standards. So there are ongoing challenges in reconciling existing standards with the evolving complexity of requirements over time. To help promote broader implementation and use of these standards, continued progress does rely on a collaborative approach and engaging various stakeholders in this space to work together in addressing challenges. Along those lines, to help, we'd like to share some key resources that we think will be valuable for those interested in genomic data sharing. Right, next slide, please. So one of the most exciting resources that we have coming up is the Sync4Genes Resources Toolkit. This is intended to promote awareness of the accomplishments of the Sync4Genes project, disseminate essential resources for genomic data sharing, and engage stakeholders like you in adopting, implementing, and integrating these resources. So our goal is to encourage increased utilization of the outcomes from the project, many of which we discussed here today. The toolkit itself, just to give a little bit more information on it, it'll provide a comprehensive summary of the findings and recommendations from each phase of Sync4Genes, so the phases that, again, that we looked at here today, as well as an informative collection of resources, as well as guidance for implementing genomic data sharing. The toolkit has been designed to support a wide range of stakeholders, as there are many, again, in this space, and underscores the importance of genomic standards and the extensive knowledge generated throughout the Sync4Genes project. It'll also highlight areas that would benefit, again, from focused efforts and further collaboration. The topic areas are going to be structured to provide an introductory understanding of the challenges as well as the potential solutions. hopefully equipping stakeholders with the knowledge needed to effectively tackle these issues and leverage the lessons learned from the project. So please stay tuned to ONC for the release of the resources toolkit. I'm sure that will be available on the Sync4Genes webpage on the ONC website. Right, next slide, please. All right, so taking a closer look at some of the core resources that we also highly recommend as an outgrowth of this particular project. So as has been mentioned many times already, the Sync4Genes program page for the latest updates, phase overviews, and detailed information on the project's objectives and outcomes. Our Sync4Genes website contains detailed information about the project, including reports from each of the phases, so you can take a closer look at the details for each of the phases that we provided an overview on, and each of the final reports and other materials that have been released along the way. Next up, we have the HL7 FHIR standard, so you can check out the homepage to learn more about this really crucial foundation for the interoperability of health data, including genomic information. Next, we have the HL7 reporting implementation guide. So this provides detailed instructions on how to implement the HL7 FHIR standard for genomics reporting, which again is a key output of the Sync4Genes project. And then there's the HL7 FHIR Genomics Implementer Guidance, and that's a resource for implementers of the FHIR Genomics standards, which provides guidelines, best practices, and technical specifications. Then there's the HL7 Clinical Genomics Work Group, which is a community working on genomics data standard development and implementation. So you can connect, and we encourage all stakeholders to connect with this work group, which focuses on developing standards for the representation and exchange of genomic data in healthcare. And lastly, as you've heard mentioned previously, the Global Alliance for Genomics and Health, or GA4GH, is a global initiative dedicated to advancing genomics research and ensuring its responsible and effective translation to healthcare. They also develop standards and help provide a framework for responsible sharing of genomic and health-related data. Each of these resources offers unique insights and tools, again, that we encourage all supporters and stakeholders of this work to take a look at, and it can really provide that understanding and application of continuing to advance genomic data sharing. Next slide, please. All right, and with that, we've reached the end of our session on ONC's Sync4Genes project, which again has significantly addressed high priority challenges in the adoption, integration, implementation, and use of genomic data. So advancing the use of this data in healthcare is a collaborative effort, and stakeholder involvement, again, is essential to its success. It relies on individuals and organizations helping to continue refining and developing standards that accurately represent the work that's being done in the genomics community. So we hope the upcoming Sync4Genes resources toolkit that we previewed briefly today, along with the, again, extensive trove of materials and knowledge that have been generated by the project over the years, we hope this will be a valuable guide. And thank you to our presenters today, and of course, all of you for joining us. We hope you found that this webinar to be informative, and please watch out for the rest of the Tech Forum sessions and workshops that ONC will be hosting throughout the year. And as always, whether it's specific to the topics that are covered today or more general, please feel free to engage with ONC via one of the available ways listed here. Although we always highly recommend the Health IT feedback and inquiry portal for sharing any Health IT related feedback or concerns that you'd like to bring to our attention. So again, thank you everyone, and have a good rest of your day.